Hello there, my name is Dorian Garrick and it's my privilege to give you a talk about livestock improvement to the Estonia Beef Breeds Association. So my job is in animal breeding and genetics. Genetics is the study of inheritance. Animal breeding is all about using knowledge of genetics to try and improve a population. So let me explain a little bit about the nature of the problem. So we have a pool of selection candidates, and these are individuals that we could choose to become parents of the next generation. In this particular example I'm showing you, I have performance for two traits. On the x-axis, I have the birth weight, and on the y-axis, I have the weaning weight. So these are two traits that might interest me in my production system. Now, each of the circles represents a different sire that I might choose to use in the future. So what you'll notice when you look at this pool of selection candidates is they don't all sit in a line and they don't all sit in one large circle, but they sit in an ellipse. So the bulls that tend to have offspring that are higher performing for birth weight, above average for birth weight, also tend to be above average for weaning weight. And the sires that have offspring that are below average for birth weight also tend to be below average for weaning weight. That's because these traits are genetically correlated. They are positively genetically correlated. So being larger at one age tends to be associated with being larger at other ages. Now this selection problem is relatively simple if I can make my selection based on progeny means where the sires have already been used. And the sort of decision that a farmer might make if they were faced with this problem would be to say, from experience, I don't like birth weights above 40 kilos, perhaps because of problems of calving difficulty, but I like my weaning weights to be as heavy as possible, perhaps over 250 kilos. So the black spots represent the bulls that have progeny birth weights below 40 kilos and progeny weaning weights above 250 kilos, and those might represent the team of sires that would be used as parents of the next generation. And if we were to use those animals as parents, what we would do in the next generation is we would move that pool upwards with higher weaning weight and a little bit to the left with a lower birth weight. So something we know about selection is we can't change the amount of variation that exists for a trait like birth weight or the amount of variation that exists for a trait like weaning weight, but we can move the mean. We can move that whole pool. We can move that whole pool to the left or to the right or up or down, depending on how we do our selection. And the reason that we see a change in performance from one generation to the next is because the animals that we've chosen as parents have different from average breeding values. So let me explain a little bit about the concept of a breeding value. So here I have a sire. This happens to be a Brayford sire, a cross between a Bos Indicus and a Bos Taurus breed that might be considered in some tropical environments. And I want to decide whether I'm going to use that bull as a father or not. Now, some of you might look at him and decide you like the look of him, or you might look at him and you decide you don't like the look of him, but you are making that assessment based on his own phenotype or physical appearance. Whereas when I look at a bull and decide whether I like him or not, I make my assessment based on what I think the performance of the next generation will be. That is, what the performance of his progeny will be. So if I think this is a good bull, it means I think his progeny will be above average. And if I think this is a bad bull, I think his progeny will be below average. So we observe the sire's breeding value through looking at his descendants. So if I generate a number of offspring from this bull and I measure them relative to the average in a particular herd, I will see that some are heavier than average, some of the offspring, and some are lighter than average. And the differences that we observe will partly be due to the age of the calf. The calves born earlier will be heavier, and some of them differences will be due to the age of the mother, because that affects how much milk production they produce. But if I've adjusted for the age of the mother and the date of the birth of the calf, I will still see variation, with some of the offspring being quite extreme, like these calves here at, say, plus 30 kilos relative to the herd mean, and some being even below average. There's one calf there at minus 10 kilos. So it turns out that if you were to do this and adjust for all those non-genetic effects, such as age of the dam of the calf and the date of birth of the calf, you'd find that the variation in performance of the offspring of that one sire exhibits more than 3/4 of the diversity you would expect to see in the entire population. Now, a lot of farmers find this hard to believe. They expect a good bull should produce good offspring and a poor bull would produce poor offspring. But when we look at real life data from these kind of experiments that occur on every farm every time an animal is chosen, we find that there's a huge variation in performance. And we can't change that variation, but what we can do is change the mean. So in this particular case, the mean of all of those progeny deviations for this particular bull is plus 10 kilos. So that tells me that this bull is likely to be above average. If this bull had had hundreds or thousands of offspring, then I would say the breeding value of this bull is twice that, at is plus 20 kilos, because I know that the genetic material of this bull is stored in the form of chromosomes, pairs of chromosomes, and only one chromosome from each of those pairs is passed on to the offspring. So the average performance of the offspring is only reflecting half of the merit of the bull. So I would say the bull is 20 kilos better than average. If the offspring of this bull is from a relatively small cohort, then I can't trust that average difference of plus 10 kilos as much as I could trust it if it was based on hundreds or thousands of offspring. And the statistical methods that we use to predict breeding values is something that's known as a shrinkage estimator. So we shrink what we observe based on how much we can believe it. In this case, if the bull had had about 80 odd offspring that had varied but on average were plus 10 kilos above the mean, we would shrink it just a little bit and say the EVV of that sire is likely to be in the range of 16 to 18 kilos. So there is a lot of very sophisticated statistics and computing that go behind the scenes of these evaluations, and the methodology that is used throughout the world was methodology that was invented by Dr. Charles Henderson while he was doing his PhD in Iowa, and was subsequently published in the 1950s, and successively there have been many advances to it. It's an approach known as best linear unbiased prediction, and while the details aren't too important for farmers in the audience, what this approach does is simultaneously estimate the effect of the age of the mother of the calf and the date of birth of the calf and the deviations of each of the animals that exist in our population, and comes up with the best estimates we can possibly get given the available pedigree and performance information as to the EBV, or estimated breeding value, of the bull. So if that bull had a breeding value, an estimated breeding value of somewhere in the range of 16 to 18 kilos, and we were to generate new progeny from that bull, then I could tell you that I expect half of that superiority to be passed on. So I would expect a new crop of offspring of that bull to average 8 to 9 kilos heavier than those from an average sire. But I would recognise that some will be quite a bit better than that, and some will be less than that. And we can't tell which of those offspring are better and which of those are poorer without buying more information about them, which we do by measuring and recording their performance. So the traditional manner of estimating a breeding value and explaining to farmers what a breeding value means is all based on phenotypic performance. But there is also a genomic explanation for what we have seen, and I'm just going to now explain that concept. So chromosomes are a sequence of chemicals known as base pairs. Cattle usually have 30 pairs of chromosomes, and one of each of those pairs of chromosomes will have been inherited from the sire, and the other member of the pair will have been inherited from the dam. A typical chromosome comprises about 100 million of these base pairs, which take 4 different forms: adenine, guanine, thymine, or cytosine. So in total, about 3 billion of these base pairs describe the animal. Now, only about 1.5% of the base pairs in those chromosomes actually represent genes that make some sort of product, like an enzyme or a hormone, which directly influences the performance of the individual. And I've represented those by the little squares in blue. And I have coloured the 2 chromosomes in the pair, one being yellow, representing the chromosome that came from the father of this bull, and the other one in orange, representing the chromosome that came from the mother of this bull. So these chromosomes, these pairs of A's and T's and G's and C's, they have a particular structure known as a helix, or a double helix. And whenever on one side of that double helix you have an A, you'll have a T on the other side. Whenever you have a C on one side, you'll have a G on the other side. So the double helix is rather like a ladder that has been twisted around like a corkscrew, with each rung representing A and T or G and C. When a chromosome is copied, if you can imagine this ladder being ripped in half, as you can see at the bottom of this diagram here, if it's ripped in half and then a soup of loose base pairs, A's and T's and G's and C's, were to bind with the broken parts of the rungs of this ladder, you would end up with 2 copies of this ladder that were exactly the same. But in actual fact, when this occurs, mistakes are made. If I was to ask you to write down the identification numbers or the weaning weights of 100 animals, an average person would make an error about 1% of the time. And the same is true when a chromosome gets duplicated like this. So a chromosome with 100 million base pairs, there would be a mistake made about every 100th base pair. So about 1 million mistakes would be made. And a common error that would be made is where one of these base pairs gets substituted for another base pair. And that's what we refer to as a single nucleotide polymorphism, or a SNP. Poly means many and morph means type. So a polymorphism just means there's more than one type. A single nucleotide polymorphism means that at a particular position, the nucleotide might change from, say, an A, which is normal, to some other nucleotide, perhaps a T or a G or a C. That would refer to a SNP. If we had 1 million SNPs being created when a chromosome was copied, that would be catastrophic. It would change the genes hugely, very, very quickly, and it would likely damage many of those genes such that they would no longer operate. So in a cell, there are genes whose job it is to go and repair those errors. And those genes are incredibly good at looking along the chromosome and identifying where errors are made and fixing them. And it will fix almost every one of them. But typically, it will allow about one new error, one of those SNPs, to be created each generation. So if you were to compare one of my chromosomes to the same sequence that I inherited from my mother or my father, you would find that about 1 in 100 million was unlike the version that I got from my mother or the father, but the result of one of these errors known as a mutation. So some of those mutations will not be in genes, and some of them will be in genes. And when the mutation is in a gene, it might possibly make that gene work a little bit better. It's probably more likely to make that gene work a little bit worse. So these genes might represent, hormones, for instance. So a hormone like testosterone, which is responsible for maleness in a bull, is controlled by a gene somewhere along the genome that has a particular sequence of these base pairs. And the hormone, when it is produced, will be a chemical compound that flows through the bloodstream and locates a receptor which is produced by another gene, and the hormone and the receptor will join together to turn something on or to turn something off, rather like a key will go into a lock, and if the key goes into the correct lock, it will be able to open the lock. So if you make a small modification to the gene, you might make a small modification to the hormone or to the hormone receptor, and that might make them bind a little easier, or might make them a little more difficult to bind. But the net effect of it will be that it might change performance a little bit in one direction or the other. So these kind of mutations have been occurring for generations, and some of them have been passed on by chance, and some of them have been accumulated through the results of natural or artificial selection. So when we talk about the breeding value of a bull, what we're talking about is the value of all of the gene effects that bull is carrying that influence the particular trait of interest. So if this bull had an EBV of 10 kilos, then what that tells me is the sum of all of these variants that that bull has at genes spread along the chromosome will sum up to 10. And some of those variants will have been inherited from his father, the yellow strand, and some of them will have been inherited from his mother, the orange strand. And if I talk about 3 bulls that have 3 different EBVs, so one bull has an EBV of plus 10, another bull has an EBV of minus 6, and a third bull has an EBV of 2, then I expect exactly the same genes to be involved in determining the EBVs of those 3 bulls. But the bull with the higher EBV is going to have more of the favourable form of these genes than does the bull with the poorer EBV. One of the things you'll notice is that even the bull with the highest EBV here, the bull with an EBV of 10, has some negative forms of gene, and even the bull with the worst EBV, the one at minus 6, has some positive forms of the gene. And this is why it is that a bull that is below average will sometimes produce above average offspring, and a bull that is above average will sometimes produce average or below average offspring. At any one particular locus, there are 3 genotypes. There are individuals that have 2 copies of the good form of the gene, and that's what I've represented here with the letter capital Q. There are individuals that carry 2 copies of the bad form of the genes, that's the little q, and there are individuals that carry 1 copy of the good form and 1 copy of the bad form. The animals which are big Q, or little q, we would describe as homozygous, because when they become a parent, it doesn't matter which of the pairs of chromosomes is sampled, it will have the same value. A big Q bull will produce all sperm with a plus 4. If the bull has a big Q and a little q, we describe that bull as heterozygous, meaning that that bull can produce different kinds of zygotes. So in this particular example, if the offspring inherit the big Q, that sperm must have carried the plus 4. If they inherited the little q, then they would have inherited the minus 4. So that particular location, or what we would call QTL, or locus, contributes to the breeding value. The heterozygous animal would be average. The big Q animal would be plus 8 relative to average, and the minus 4 animal would be minus 8 relative to average. So what we would like to know is where all these genes are that influence performance and what the value of those alleles are. And the way we might determine that is using a technique known as regression. So in regression, if I wanted to know how fertilizer affected the growth of my grass, I might put no fertilizer on, or 1 bag of fertilizer on, or 2 bags of fertilizer on, and measure how much grass grew. And I might want to know how much more grass grows for each unit of fertilizer that I apply to my paddock. That's the same concept as we are interested in here. On the y-axis, I would have the true breeding value of a bull based on progeny test from many offspring. And on the x-axis, instead of talking about 0, 1, or 2 bags of fertilizer, I talk about the effect of having 0 good big Q alleles, or 1 good big Q allele, or 2 good big Q alleles. The slope of that line represents the average effect of that particular allele or that form of the gene, and represents how much the performance improves when an animal carries one of those favorable forms of the gene. So this concept has been around for decades, but really it has only been revolutionized when a company called Illumina invented something called a SNP chip. And here I show you several different kinds of SNP chip. The first kind of SNP chip that came along was one that was referred to as a 50K, because this SNP chip could determine what particular SNPs existed at 50,000 different positions on the genome, all from one reaction. And this thing here, which looks like a microscope slide, represents a whole lot of spots where a different sample goes from a different animal. So it might be that one slide can simultaneously interrogate perhaps 12 or 24 different individuals at as many as 50,000 or 700,000 different locations in the genome. So if I was to apply one of these chips to my bulls, what I would get back from the genotyping company is that at a particular location or place in the genome, some bulls are heterozygotes. They would be referred to as AB. Some individuals are homozygous. They might be referred to as BB. And some individuals are the alternate homozygous. They might be AA. Now remember, there are 100 million base pairs in a typical chromosome, and only about 1% of it, 1.5% of it is in genes. So many of these loci that are reported on the chip will be in the regions between the genes, what we call the intergenic regions. They won't be the genes that actually have the effect on increasing or decreasing performance. What we are hoping might happen is that the SNP is in what we call linkage disequilibrium with one of the real genes that is having an effect in this region. So in this case, I show you the A allele is associated with the +2 variant, and the B allele is associated with the -2 variant. So when an individual is homozygous for the AA variant, in this example, it's also homozygous for the +2. And when it's homozygous for the BB variant, it's also homozygous for the -2, somewhere nearby. And when it's heterozygous for A and B, it's heterozygous for the -2 and +2 form. So in real life data, what I might get is some breeding values that I could regress on the number of copies of the B allele that occur at one of those 50,000 positions in the genome. And if that particular position was in high linkage disequilibrium with one of the genes that had a real effect, then the AA individuals would mostly be the individuals with the lower breeding values, and the BB individuals would mostly be the individuals with the higher breeding values. But if the linkage disequilibrium is not perfect, there will be some mistakes made. So some of the AA individuals might be big Q, little q. Some of the BB individuals might be big Q, little q. And maybe some of the AB individuals will be big Q. Some of them will be little q, but most of them would be big Q, little q. Then when we do the regression, the slope we will get will be not quite as steep as we would have got if we could have observed the actual gene. So what we do when we do a genomic analysis is, you could imagine walking along the genome and at 50,000 different positions asking the question, just like how much extra grass goes for an extra unit of fertilizer, we would ask, how much more breeding value do I get for an extra copy of the B allele at this particular SNP locus? If I go to the next locus, which is in lower LD, I might find that there is no signal. That SNP doesn't pick up the effect of any particular favourable allele. So as I walk along the genome, I'm going to find some effects in some places, and I'm going to find other places with no effects. Now, this technology was first made available to the public through a company called 23andMe, and the company was called 23andMe because in humans, unlike cattle, we have 23 pairs of chromosomes. And they did this particular experiment in relation to Alzheimer's disease. They had people with Alzheimer's and people who did not have Alzheimer's. And they did a regression of the number of copies of the SNP on the presence of that disease. And they found one particular region of the genome near to a gene called ApoE was associated with Alzheimer's disease. So here are the results for me at two SNPs, RS7412 and RS429358, which are located in the ApoE region. They can determine the genotype from the SNP chip, and they can determine that my odds of getting Alzheimer's disease relative to other Europeans is about 0.67. Doesn't mean I won't get Alzheimer's because it's not entirely determined by genetics, but it says that relative to other people whose genotype is unknown, I am less likely to get Alzheimer's because I am carrying forms of the genes that are associated with reduced risk. Now, the same exercise, this what's known as a genome scan or a genome-wide association study, has been done for many diseases. Here are the results for coronary heart disease. And in the case of coronary heart disease, they found lots of different positions in the genome that tended to be associated with positive or negative variants. The likelihood of getting heart disease is determined by genetics with what's called a heritability of somewhere between 39 and 56%, according to the publications reported in this website. And what that means is for an average person in the population between the ages of 45 and 79, about 47 of them, about 47%, 47 out of 100, are likely to develop heart disease between 45 and 79. But people with a genotype like me are more likely to get it. We are likely to get it with a probability of 55%, or 55 of us out of 100. And they have come up with that number by adding up the positive values and the negative values of all the variants that have been shown to be associated with coronary heart disease. And on this particular graph, each of the columns represents a different location in the genome, and where the column is red, it means I have the unfavorable form of the gene, and where the column is green, it means I have the favorable form of the gene. So just like those bulls, I have some favorable and some unfavorable forms, but I have more unfavorable forms than favorable forms, which is why I am more likely than an average person to get heart disease, because I carry a genotype that is unfavorable. Now, in these particular cases with humans, they only use significant, validated genome-wide association studies to do the predictions. Whereas when we do these things with animals, we tend to use every SNP that is on the chip. The theoretical basis for accuracy using this methodology has been developed, and one of the early papers doing that by Mike Goddard and Ben Hayes, published in 2009, showed how many individuals we needed in what they called the training population. So that's how many individuals they needed when they did the regression of breeding value on number of copies of the SNP allele to get a certain level of accuracy. So I've shown you here the graphs with green, orange, blue, and red represent traits with different heritabilities. A high heritability means there is a strong association between phenotype and genotype. A low heritability means there's a weak association between phenotype and genotype, and that might occur when there is a lot of chance effects on performing above or below average, rather than just effects determined by genes. So to explain 20% of the genetic variance in breeds such as Holsteins and Jerseys, we would need a correlation of 0.43. 0.43 times 0.43, R squared, would be 20%. According to this graph, for a high heritability trait, we'd need 1,000 animals in our training population that have genotypes and phenotypes. If we wanted to explain 36% of the variance, we'd need a correlation of 0.6, and that would require 3,000 animals. So reliable prediction requires a large training population of genotyped and phenotyped individuals to work out which genotype regions are favourable and which ones are unfavourable. In the human context, they used to aim on getting 100,000 animals in such a population. Nowadays, they often aim at getting more like 1 million animals. So how well does this work? One of the best examples of genomic prediction in livestock is from a paper back in 2010, published by one of my colleagues, Anya Walk, in chickens. And here we show you on the x-axis some different traits in chickens, in layer chickens. So PD is egg production, PS is puncture score, it's how easy the egg breaks, YW is yolk weight, AH is egg height, AH is albumin height, E3 and EW are egg weights, and CO and C3 are to do with the colour. And the black line that goes up from the left to the right represents the accuracy of prediction we can achieve when we do selection using phenotypes and conventional pedigree relationships. The red line, which you'll notice there is superior for all the traits, represents how well the prediction is done when we use genomic relationships, or we use the same kind of prediction as I just explained to you that are used in humans for Alzheimer's disease or for coronary heart disease. And the purple line there represents what happens if we only used the parent average genotype and didn't account for the chance sampling, what we refer to as Mendelian sampling, that results in some individuals being better than we would expect relative to parent average, or worse than we would expect relative to parent average. The impact on accuracy from genomics depends on how well the genomic prediction is correlated with the breeding value. So a genetic correlation of 0.3 would represent a not particularly strong relationship. And this shows you how much the accuracy of prediction improves when we add genomic information on top of pedigree information against the accuracy of the bull. So in this graph, if a bull has an accuracy of 0.6 or 0.8 or 1, It really refers to a bull that's been progeny tested with a lot of offspring. Then having a genomic test on this bull is not going to provide us with more accurate information than we could get from just having the pedigree information. But if we have a very low accuracy for the EVV of a bull, we will gain a lot of information if we add genomic information. And in this example, we might go from 0 up to an accuracy of about 0.3. The next graph shows you what happens when we have a more accurate test. And here you'll see we get a much larger bump in accuracy when we add genomic information on. And that genomic information will add information unless the bull is very, very reliable, such as a bull which has been progeny tested with thousands of offspring. So we get a much better return on genotyping investment if we have an accurate genomic prediction. But to have an accurate genomic prediction, we need a large training population of individuals with genotypes and phenotypes, which means we must have invested in a lot of historical performance recording and genotyping of historical animals. So, in summary, genomic prediction exploits advances in quantitative genetics, in statistical genetics, computing, molecular biology, and bioinformatics. It is the basis for some aspects of personalized medicine, so it's being used in humans. It will revolutionize animal improvement programs, but to different extents in different industries. It's been a much easier technology to adopt in the dairy industry than the beef cattle industry, because dairy bulls are very valuable and you can't measure milk production in the bull themselves. You have had to progeny test the bull to identify a bull with an above average breeding value, and that costs a lot of money and takes a lot of time. In beef cattle, it's hard to get a return on the same kind of investment, so it's taken longer to develop training populations, and they're not nearly as big as they are in dairy cattle. So the accuracy is not as high in beef cattle as it is in dairy cattle, but it is improving all the time. Livestock improvement results from using above average individuals as parents of the next generation. And in order to do that, we need to decide which traits need to be improved. That's what we refer to as the selection objective. And typically, there will be a portfolio of traits that will be simultaneously important to us in selection. We need to record pedigree and performance information. We need to do this to develop training populations, and we need to do this to develop traditional progeny test kinds of estimated breeding values. But with genomics, we can assure that we have accurate pedigrees, and we can get improved accuracy in young animals. We need to evaluate animals for the traits that matter, and this involves an evaluation system. So we need a database to store records, and we need a system to do the BLUP kinds of evaluations, to take all the pedigree records, all the performance records, and all the genomic information, and provides the EVVs of the bulls. Thanks for your time. Goodbye.